Am I audible, madam?
Yes, sir. मैम बना दिया मैंने होस्ट सर। क्लास में जी Am I audible, sir?
Yeah, I'm audible. Should I start, madam? Shall we start?
Yes, sir.
Yeah.
Yes, sir. Good morning,
So,
everyone.
madam, give me sharing access. So,
Okay, sir, you can share. Yes.
but it is showing sharing is not turned on. Send
Let
request.
me. Okay, let me turn.
I
Okay,
sent
okay, sir.
request.
Yes, I am allowed.
Yeah.
Good morning, everyone. It gives me immense pleasure today to welcome you all to the Gurukul Pacific Expert Lecture organized by Pacific College of Pharmacy, Pahir University. A very warm and special welcome to our esteemed resource person, Dr. Niradi Dinesh sir, who is CEO and founder at Niradi Biosciences Private Limited at Hyderabad. Sir, with your research experiences at prestigious institution along with your contribution to pharmaceutical biotechnologies, and infectious disease research, scientific publications, and patents, we are truly honored today to have you with us and grateful for your valuable time and guidance. I also extend a warm welcome to our respected President Sir and Dean Sir, along with all the faculty members and students joining us today. Our today's topic is Future of Pharma Research. Artificial intelligence in drug discovery and development is highly relevant in today's rapidly advancing pharmaceutical field. I am sure this session will be highly insightful and inspiring for all of us. Now, not taking much time, let me invite I'm Dr. Niradi Dinesh sir to kindly take over the session and enlighten us with his valuable insights. Thank you. Welcome, sir.
Yeah, thank you so much. Thank you for inviting us. And thank you for giving me the opportunity to interact with young minds. And thank for the university management and all the faculty of Pharmacy College and the director, sir, and president, sir. So thank you. And it is a privilege to interact with the current generation and talking about AI, how it will be useful for the current pharma students, especially. So we will discuss for another 1 hour. We will be talking different topics which will be useful for your career and ongoing projects in worldwide in pharmaceutical industry. So I'm Dr. Dinesh. So I'm a CEO and founder from Niradi Biosciences Private Limited. I did my PG PhD from Naipur Mohali, so All India first rank. Then I joined in ICMR NIN, National Institute of Nutrition. Then CSIR CCM, I worked for various central government projects like COVID vaccine discovery and COVID diagnosis, and also filed 6 patents, US patents, and also worked on drug repurposing. Okay, so developed new molecules for the treatment of various diseases, like especially infectious diseases. So with this background, I will try to communicate to all of you. So with my personal experience, and I'll also tell you what is happening in the current pharmaceutical industry, right? So yeah, so our main goal for this session is demystify AI and show how it transforms the traditional pharma pipeline. So how the previous drug discovery changed and went into different level and in a faster mode by using AI. So how it is helping the pharma industry to discover the drugs in a faster mode. And we will also try to understand from target identification all the way to post-marketing surveillance. So now the life became very easier. So we can monitor, we can access each and every department in a minutes by the FDA, by the CDSCO, by the academicians, and from pharma industry. So this is the main objective for this session. We'll try to cover these two. And first we'll discuss the pipeline and versus AI paradigm. So what is the older style of drug discovery? Now, how the AI is helping in drug discovery in a faster mode. And then we will discuss some of the artificial intelligence techniques, algorithms that is giving more value addition to the drug discovery in a value chain and supply chain. And then we'll also discuss some of the case studies, okay, happened recently, how the drug discovery changed from years to months. Okay, so in recent 2022 and 2026. And then we'll further move to some of the regulatory and ethical and IP challenges by using AI, and then future perspectives and what are the opportunities available in pharma industry throughout the worldwide. Let us start with the traditional drug discovery pipeline. So if we talk about the traditional drug discovery, it will take 10 to 15 years of time. Okay, 10 to 15, it used to take 10 to 15 years of time. And lots of people like pharma students like you and scientists and engineers, right? And management, so many people are investing their energy in drug discovery every day. They will come to lab and they'll make available lab to run to accessible to everyone. Okay, so lots of people are involved. And then the cost of the drug discovery was, it was written here $2.5 billion, but now it is changed. It is costing almost 3 to 4 billion US dollars. Okay, so the cost was very high. And then coming to the success rate. So the olden traditional older approach of drug discovery, the success rate was only below 10%. So the people used to start with the lakhs of molecule and they'll end up with the 10 lead molecule. Okay. So from lakhs to just they'll end up with the 10 lead molecules. In the 10 lead, one may be successful, may not be successful. So only 10% chances. Okay. So this is the older style of drug discovery. But now what happened? The AI changed so much. Okay. AI influenced our life. Yes or no? Do you believe this? Is really AI influenced our life? Yeah. So I think you can chart. Yeah. Is a chart accessible, madam, for them? Students can they can interact, or shall we continue?
Yes, sir. Chart is not sharing on the screen.
Fine, fine, no issue. Okay, fine, fine. It is available. Huh? Right. Okay, fine. So we'll continue. Yeah. So now the AI changed so much in our daily life and also drug discovery and pharmaceutical industry. So have you heard about this Erum's law? Okay. So if you reversely, if you talk this Moore's law. So Moore's law means, so what the statement, so whenever in the olden days, the drug discovery was very slow. Okay. So the drug discovery was very slow. But when the computer entered into the drug discovery, computer entered into the pharmaceutical industry, it became cheaper and it became more accessible. That is Moore's law. If you read in a reverse way, it is Erum's law, right? So now we are easily accessible to drug targets. Okay, drug target identification worldwide. So you can access any drug target by just sitting in the computer. But in the olden drug discovery, it is not possible. They used to read the literature, go to library, pick up the articles, and read the patents. Then they used to get some data, and sometimes there was a duplication. So some people are working in India in the same research, and some people are working in abroad also same. So it used to become duplicate. So but now it is avoided. because of AI. So the data is accessible to everyone, right? And then how? So before AI era, so there was a serendipity and high throughput screening, right? So serendipity means people are working for some reason, some purpose, and suddenly the activity of that particular drug candidate arised with a new activity. Okay, so like that, there was a serendipity area. Okay, so just people are not discovered the drugs by purposefully. So there are so many serendipity drugs, you aware. So like sildenafil. Okay, so this is not the drug which is discovered for the purpose, but they are doing some trials and they got some new function, and then now that is in the market. Okay, like that there are so many like antibiotics. So antibiotics are not purposefully discovered, right, by Alexander Fleming, right? So he is a scientist working on microbiology lab, and suddenly one day he saw some zone of inhibitions. Then he extracted that particular agar extract. Then he went for the chemical analysis. Then he found antibiotics. So that is all serendipity and high throughput screening. But now, after AI entered into the pharmaceutical industry and the drug discovery, now it is a rational design. So we are starting the drug discovery with a purpose. There is a rational. What is the problem? Okay, first we are starting with the problem. Then we are going for prediction modeling. So how it can work, whether it will work or not, what kind of molecules are required for this particular disease, for this particular target. So now so much is changed. So there is a change in attrition by failing faster and cheaper in silico. So in the olden days, they used to screen thousands of molecule, but at the end, they so many molecules are failed. They are not in a that is showing maximum toxic effect or less activity or bioavailability problem, right, or solubilization problem at the end. By spending so much of energy, then people used to throw that molecule into the dustbin. But now, before going into the wet lab, you can analyze that particular molecule in every stage. You can assess the toxicity, you can assess the activity, you can assess the metabolism, you can assess the absorption, distribution, everything you can assess by using in silico. So that's how the AI changed the drug discovery in a faster mode. So what do we mean by artificial intelligence in pharma? So yes, AI involved in everywhere. AI, the mobile also working on AI. Okay, and even machines or flights and everybody using now AI. But what is actual AI in pharma? What we should know if you want to apply this AI into pharmaceutical industry. Number one, machine learning. So in machine learning, so what we should understand is pattern recognition in historical data. Okay, so if somebody studied about some drug target, okay, somebody studied some drug target or drug candidates. So by using AI, you can analyze the historical data. Okay, so that's how machine learning will help us to understand the reported data, published data of various drug candidates or diseases. Then the other one is deep learning. So here the neural network involved in deep learning for images and sequences. Suppose if you are working some genetic disorders in India, okay, so you can find out this particular genetic disorder information of other countries as well, how it is happening over there, what is the sequence in that country, what is the sequence in our country, what are the changes. Okay, so you can understand in deeper about that particular genetic disorder. And the other one is generative AI. So this is creating novel molecular structures. So in a short span of time, so you just give the target, give the receptor structure to the AI, and then that will give the modeled structures. Okay, modeled molecular structures which will fit into that receptor. So that made life easy. And then another interesting AI platform, that is NLP. So this what it will do? So it will do the literature work. Okay, so for the in the drug discovery, so 1 to 2 years it used to take for the literature in the initial olden days, because at that time the computer is not available. People used to go to library, travel from one place to another place, the central library, and they used to search all the articles, hard copy articles. Then they used to do the literature review. But now you can sitting in your room, single room with the AI and with a computer, then you can do the literature review of in the worldwide. Okay, so that's these are the four major things which will help in pharma in drug discovery. So now let us understand what actually is happening, right? So there are the only important use of this AI handling data. Okay, so the giant data is handled by the AI with the accuracy. Right, in the olden days, all people used to sit and after experiment, they used the data analysis by taking the hard copy reports. Okay, that used to take so much of time. That's why the drug discovery took 15 years. But now the time has reduced a lot. So I will tell you some of the examples how they discovered in years, how they discovered in a months, right? So here AI is AI is only the part of the drug discovery. It is not complete drug discovery. Always human interference always required. Now people are talking about AI will reduce the jobs, AI will cut down the employment. No, it is not. AI always improve. AI always impact the drug discovery by with the human intervention only. So what it will do actually? So AI will help to identify the chemical structures. Just it will read the data. That is smiles. So suppose if you want to work one drug in your project in your college, so faculty gave you some title. Okay, so what you have to do, you cannot just search with the title in the Google. It will give some minor information. But if you go and type the drug name and the smiles, so the smiles language is represented, the drug names represented in smiles language. AI will not read your text information. Okay, you must communicate with AI with the smiles strings. Smiles is the names for the drugs. Okay, this is the language, right? And then biological, so genomics and proteomics. So all the genomic data, the now the human genome, complete human genome, which includes 25 to 30,000 of human genomes data, genes data is available now. It is accessible to everyone. Okay, so in 2007 2009 they completed whole human genome project, and every day it is getting updated. Now you can access everyone's data. You can access Africans genes, human beings genes. You can access Americans human genes, okay, by this genomics and proteomics data. And then clinically, now EHR is helping the hospitals to understand the patient's patient treatment and their consequences also by using softwares, right? And then imaging, MRI and histology. So now, if you take 1,000 images and if you submit to AI, okay, so that will give the differences. That will identify accurate differences. What are the major changes happen from test to control, okay, or reference or sample. So that's how the shift helped the drug discovery in a faster manner. Now it is a fair data and easily findable. Okay, you can access if you want some population. Suppose if you are in clinical trials, phase 1, phase 2. Okay, if you need some parameter, you just find that parameter. You will get all the data related to that parameter from the giant big data. Lakhs of parameters are there, but from the lakhs of parameter, AI will pick that single parameter and that will give you the information, right? It is findable and accessible and interoperable and reusable. Anyone can operate. So in the olden days, so one person working in the A shift and the second person who is coming in the B shift, there might be changes. So this person handling is different and the first person handling is different. There might be errors, data errors, okay, experimental errors. But now the every experiment is monitored by the AI. So whatever the parameters which he used in the A shift, that is directly translated to the transformed and transferred to the B shift person also by the AI. No change in the parameters. So that's how it is interoperable and then reusable also. And then coming to there are two different techniques here. The one is predictive and then generative AI. So how predictive AI will work? So if you come up with a bunch of molecules, then this predictive AI read all those molecules and it will tell you the information whether this particular molecule will bind to this receptor or not. So that is a predictive AI. Okay, predictive artificial intelligence, right? And this will tell you the classification and the regression on known data, right? So if you know the receptor structure, if you know the chemical structure, then that will see whether it will fit into or not. Then generative data, what it will do, if you do not have any molecule, if you do not know any information, then it will design the molecule that binds to the target with these properties. So this is called de novo molecular design. If you do not have any molecule also, you can start drug discovery and it will give you the lead molecules. It will generate the molecule which will go and bind to that particular receptor, right? And then the other thing is you must understand biological data. So you must know, you must try to understand the biological data also, especially DNA, RNA, proteins, right? So there are so many supercomputers now handling this data because this is a huge data. So the human genome size is 3.2 billion base pairs. Okay, thousands of genes are present. Okay, if you want to understand a single drug effect on the human body, then you must understand how the drug is affecting the other genes, which we are not targeting. Okay, so in that way you have to understand metabolomics, which you have to understand proteomics. Now omics entered into the drug discovery. Even though you are using one drug for cardiovascular problems, but that might be affecting the other systems also. You must study the other system. You must look into the other systems of the body also. So that's how the biological foundation models now helping to understand the whole data in a systemic manner. Okay, so this is the main. There are so many supercomputers like LLMs. We call LLMs large language models trained on biological sequences. So DNA, RNA, and proteins. So once you train your particular algorithm, particular artificial intelligence, so that will remember that particular sequence. And whenever you encounter any issue about that, then it will give you the information, right? And so here the older traditional methods. So as we discussed, only that success rate is only 10%. Okay, and the clinical failures almost 60% are clinically failure. They used to fail clinical studies, phase 1, phase 2, phase 3, because lack of efficacy and wrong target choosing. So yes, so in vitro and animal models that used to give very good data, very good activity, but when we entered into the clinical trials, then almost 60% of the drugs used to fail in the clinical trials because of efficacy, because of absorption, because of solubilization, because of so many other things. That is a traditional method. But now if you are able to understand all these problems by using AI before entering into the wet lab, then it will save our time. Okay, it will save our energy also. Now, so multi-omics, as I'm telling, omics now entered into the pharmaceutical industry, right? So here we are studying about genomics. So if you want to do one drug discovery, you must understand the gene level of that particular receptor. Okay, what is the particular gene, how it is interacting, how the gene is expressing. Okay, and how you must understand transcriptomics. So while DNA to RNA, the transcription is happening, so how much rate it is transcribed, right? So you must understand that level, genomics level, transcriptomics. Then you understand the proteomics. So you understand how the protein-protein interaction is happening inside the body. And same replica we can make in the computer also. We can study the protein-protein interactions. Then metabolomics. Metabolomics means, so the older style of drug discovery, they used to take target one particular protein, and they used to hit that protein with the different molecules. They used to maximum study happening between drug and the receptor. Okay, if it is binding more, and then they go for animal studies, and then they go for the drug discovery. But now in the inside the system, you can create entire metabolic pathway. Suppose example, if you talk about the cholesterol synthesis, okay, if you talk about cholesterol synthesis start from acetyl CoA. Okay, from acetyl CoA to, there are 20 to 30 steps will be there, and then the final cholesterol synthesized. Okay, so there are so many reagents involved, steps are involved, enzymes are involved. Okay,
Excuse me,
you
sir.
are targeting one enzyme, that is HMG-CoA reductase. As we know, that
Excuse
is
me, sir. Hello.
statins we use in hyperlipidemic conditions will inhibit the HMGR enzyme. So now what we can do, we can create entire pathway in silico from acetyl CoA to cholesterol. Okay, then you give some drug, do you disturb one step. So by disturbing one step, then it will also give the information whether that other remaining 20 30 steps are disturbing or not. So that is called metabolomics. Okay, so we can study entire metabolic pathway information in the in silico before going into the wet lab. So here, what are the main problem? Okay, so what are the main graphs involved in the drug discovery? So first, the all drug discovery pipeline you all know. So first one is drug candidate, then we'll go for the target, then pathway, then disease,
Hello,
then
Dinesh
therapy.
sir.
Okay,
Hello,
here the important
Dinesh
link
sir.
between the one step to another step. Okay, and that is also minimized with the drug repurposing. Drug repurposing means, so already the drug is present for one particular disease in the market. Okay, but now the same drug will also useful for the other diseases. Like in my some of my projects, I worked on drug repurposing, like some of the antidepressants, like drugs like ketanserin. Okay, ketanserin is an antidepressant, but when I tried the drug in the anti-leishmanial, it is working beautifully. And one more drug, atorvastatin. Atorvastatin is a hyperlipidemic drug, okay, which is inhibit the HMGR reductase in the human body. But the same drug, when we studied in leishmania, it's a Kala-Azar infectious disease, Leishmania donovani. So this drug is inhibiting the HMGR enzyme of Leishmania donovani, which is a causative agent for leishmaniasis, Kala-Azar, in nanomolar range, where in our case we are using mgs for hyperlipidemic condition. But when I studied with this drug, same drug, in drug repurposing, then I got beautiful results, that is working, that is killing the parasite in nanomolar range. That's how drug repurposing is helping a lot of energy, and it is done by mapping millions of relationship across the entire biomedical knowledge graph. So it will study what is the structure of the HMGR causative agent, what is the structure of the human HMGR enzyme. So it will read in-depth information and identify the differences. So that's how we can make drugs easily by using drug repurposing. Then the another important thing now, everybody, all the companies, like major companies, they are focusing on cancer. So that is
Hello,
CRISPR
excuse me, sir.
screens and machine learning. Okay,
Hello, excuse
this
me, sir.
CRISPR technology now is in a boom area. So in
Good
the worldwide,
afternoon.
people are working on this CRISPR, especially to work on the genetic disorders, not only genetic disorders, cancers, okay, and so many other diseases, right? So what here the CRISPR will do? So it will guide the particular Cas protein and it will go and replace the abnormal gene. Okay, so that is guide RNAs. Here we are taking the help of guide RNA. So what we'll do, we'll synthesize the guide RNA, we'll make guide RNA specific to the patient. We cannot use same guide RNA to everyone. We cannot, same CRISPR will not work to all patients. So individual personalized medicine, precision medicine is going to enter. So you have to work the sequence which is present in the patient's gene. Okay, so that's how CRISPRs are going to help in the drug design and especially gene therapy. Okay, so by replacing the abnormal genes, right? So this is how the AI is helping in a beautiful way to more to get more information on precision medicine, personalized medicine. Then other important information about AI in drug discovery is validating undruggable targets. So in the olden days, it is not possible. Okay, so if the receptor or the particular drug target is accessible to the drug, then we used to do the drug discovery. But now we can design a molecule, okay, which will enter inside that particular receptor and go and bind. Okay, like nuclear receptors. Okay, so in that way we can find undruggable targets also. Target, like there are some example like crass or once considered undruggable, but now artificial intelligence changed, that became druggable target. Okay, so this is how the AI is helping in making undruggable target to druggable targets. Okay, and then the structure function product, the structure function relationship. So in the protein structure, once you know the protein structure, then you can predict the function, and also that function you can predict in a physiological environment system, right? So the structure function prediction used to do by this various techniques like X-ray crystallography and cryo-electron microscope, and this used to take so much of time. But now you just go to PDB, okay, you get the protein, particular your protein structure. Okay, and then you can use so many in silico models to know the function. Okay, to know the function. And then AlphaFold revolution. So this is one of the greatest revolution in the recent years, especially in 2026. Okay, now we can predict not just by static structures, but also if we look at the protein structure, by looking at the protein structure, the AlphaFold, they made the beautiful drug molecules which will really fit into the particular protein. So by just looking at the protein structure, you can design the molecules. You can go for the dynamic studies, and that will give the information uh, which will fit into the drug molecule, right?
Excuse me, sir.
Predicting protein ligand interaction. Okay, predicting
Please
the
share
protein
your
ligand
slides.
interaction. So by using AI, so we can study the interaction at molecular level. What kind
Please
of
share your
interaction it is, van
slides.
der Waals forces, how the drug is binding to the receptor, whether it is happening with the covalent bond or van der Waals interaction, some electronic reactions or chemical reactions are happening. So
Dr.
that's how we
Dinesh,
understand
Dr.
protein
Dinesh,
ligand interactions in a molecular level. The affinity
please share
between
your
the drug and
slides.
receptor is studied in molecular level. So that will help us how much affinity it is, how much active it is, and with that information we can also assess the toxicity also, right? And used to take in within hours. But this information we used to get in the olden days, it will take days to years. And now it is also helping in antibody engineering, right? So mainly, so recent advancement in antibody engineering, now we can synthesize antibodies. But in the olden days, we used to give the antigen to the animal models, and
Excuse me,
then
sir.
that will take almost months to develop antibodies. So after developing antibodies, then we used to collect from the sera and then we'll market. But now the AI is completely changed. Just if you give the sequence of that particular gene or protein, okay, then people will synthesize the antibodies, right? And humanization. So the antibodies which we collected from the animals, sometimes they may use to react. Sometimes they used to react, and there might be chances for allergic reactions. But now what we are doing, so we are directly synthesizing the human antibody itself. Okay, human antibody, and by using in vitro systems, so that will reduce the immunogenicity problems. Okay, immunogenicity problems by humanization, that is happening with the help of AI, artificial intelligence. And there are certain limits. Okay, so in the olden tradition, so there are only 2 million compounds. Okay, maximum 2 million compounds they can screen. Okay, that is the physical HTS library ceiling. So 2 million compounds they can screen, not more than they go. Okay, but now here by using AI we can use n number. Okay, so we can screen. Okay, so billions of molecules started with screening.
Hello. Excuse me, sir. Sir, your slides are not visible. Sir, your
Okay,
slides are not visible. Kindly share the PPT on the screen also.
1 minute, 1 minute.
Yes, sir.
Up.
Kindly share your screen.
My slides are not moving, madam.
Yes, sir. Your slides are not visible. Kindly do the screen sharing.
Wait, wait, wait.
Yes, sir. Yes, sir. Now it is visible. Yes, sir. Yes, sir. Now it is visible.
Now visible? Yeah.
Yes, sir. Now visible.
So full screen, it is fully visible or what?
Not full screen. Yes, sir. Yeah, yeah. That's fine. You can zoom it out, sir. Yes, sir. Okay. Now you can go ahead.
Confirm.
No, it, it is
Now
not
it is.
showing now. No, it
Now
is
it is.
not showing now.
Now it's not showing. Wait.
Only your video is showing, not the slides.
Okay. Wait. Now it is showing.
No, sir.
Wait. Share option. It is disabling. I don't know why. Okay.
Yes, sir. It is showing. Now it is showing.
Okay. Fine.
Okay, sir. You can continue.
So now we can screen billions of molecules, okay, with the using AI. So not only millions, but initially in the olden days, just we have a limitation, not more than 2 millions. But now we can use billions of molecule. Okay. So now coming to the AI docked molecular dynamics. So now we can understand the dynamics of protein and drug interactions. Okay, how it is behaving inside the body. Also, we can mimic and we can understand the dynamics of that particular drug receptor interactions inside the computer by using computer. Then we will get to know how much affinity it is binding, how many molecules are interacting, how many number of receptors it is binding. So like that, we can study entire information about molecular dynamics also by using AI. So this will give so much information in drug discovery. And then another important area of drug discovery is generative chemistry, right? So that is designing the undesigned. So initially, some of the molecules we are unable to design, okay, because of the limitations. But using AI, so we can become a creative chemist also, right? But you can design a proper structure related to the drug receptor, and which will bind with a high affinity. So that's how AI is helping in the chemical structure design also. That's what I am discussing. Smiles. So these smiles. So if you want to know some drug, if you want to understand some drug structure, so first you will get the smile structure of that particular drug. Okay, suppose ibuprofen. So you just type the drug name ibuprofen and smile structure. You will get the structure. So with that structure, you work in the AI. So if you give normal word of ibuprofen, AI will not identify that particular ibuprofen word. But if it is a smile, then it will identify. Right? That is a language, chemistry, and chemical information stored in the smiles form. There is a language for AI, right? And then some of the molecular graphs like atoms as nodes, bonds as edge. So these are the different terminology you must use in AI. You must able to understand the terminology. Then only you will get to know the mechanism what is happening in the AI. Okay, and then third point. So there are some clouds. Okay, cloud database we are using to handle very big data and to handle 3D structures. Okay, so these are also very important in the in the drug discovery by using artificial intelligence. And once we are selecting one drug, okay, so we can study multiple parameters. Like you can study potency, you can study selectivity, how much specifically it is binding to the single receptor, or whether it is binding to other receptors as well. So you can study the selectivity, and we can also study the solubility. This is the biggest problem. Once we get to know, once we will work in the preclinical level with some solubility, with some solutions, then that might not be working in the human phase. Okay, solubility problems. When we are going to formulation level, when we are trying to make one formulation, then solubility problems used to arise. So but now we can get to know the solubility information, and we get to know the permeability information, and then stability studies also we can do. So this is how the AI helped in every stage. Okay. So now coming to the other important area of drug discovery is peptide. So now peptides are becoming a drug candidates. So initially the drug candidate size was huge, but now the pharmaceutical industries are working with the peptides, small molecules. Okay, small minimum number of amino acids, small chain of amino acids that is going inside the body, acting at that particular specific receptor. Okay, those are peptides. Peptides are becoming drug candidates. Okay, so then especially we can take the example of mRNA sequences. Okay, so these mRNA sequence we are finding out and how much expression it is changing in one patient to another patient, and in the drug discovery also we are as we discussed, we are working in a transcriptomics also we are checking how much expression it is happening. So now the drug treatment styles has been changed. So in the olden days, the doctor used to write, okay, if you are suffering with a fever, and now Dolo 650 mg. Okay, like paracetamol 650 mg. But now the era is changing. Okay, the treatment is changing. Okay, now we are going to enter personalized medicine, precision medicine. So how much fever you have? Just I'm telling the example. If you take the example cancer, so because I'm working on a cancer, I can tell you very good way to how the treatment is working. So suppose if cancer, one patient is suffering with the cancer, okay, you cannot write a traditional dose to the cancer patient, right? Based upon his physiological parameters, based upon his biochemical parameter, how much creatinine is there, how much blood reports are there. Based upon that, you have to optimize the dose and you prescribe him. You cannot give a standard dose for patient, because 650 is a common dose, but 650 is not required for everyone. If it is having a mild fever, you cannot give 500 mg. Okay, right. So in that way, the optimization is required now. Even CDSO, the regulatory authorities, FDA, EMA, they are created beautiful softwares which is useful for the doctors. You just enter the physiological and biochemical parameters of the patient, then it will calculate the dose and you write that particular dose. You cannot blindly write the drug, particular prescribed the traditional way prescribed drug dose. Okay, and some of the other important interesting molecules like lipid nanoparticles, right? So these LNP formulations are helping us to make CAR-T. Okay, so the olden days, so in the previous, if you go back to 5 years or 10 years back, so these so like vaccines, okay, like some of the gene therapies are made by using viral vectors. Okay, because virus is having more more virulence. Okay, it will infect very faster and it will take the molecule in a faster manner. But at the same time, if you are inserting virus into the body, and that will cause problem. Okay, that will cause problems also sometimes, and that will integrate into the human genome. That is the biggest problem if you use viral vectors. But in place of viral vectors, now the industry is focusing on lipid nanoparticles. Just you take that particular gene and you encapsulate into the lipids. Okay, then send into the body. So it will the bioavailability problems are minimized. Okay, and the non-specificity is minimized, and the delivery is also similar to the viral vectors. And the other thing is cyclic peptides. That's what we are discussing. So now peptides are becoming drug candidates by we are designing peptides. The scientists are designing peptides by looking at the receptor structure. Okay, so receptor sequence, what is there in that receptor sequence? And similarly, you design the peptides, small molecules that will go and bind to that particular receptor. Okay, and then which we are discussing in the last 50 to 100 years of drug discovery, just only ADME. Okay, absorption, distribution, metabolism, excretion. But now with the AI, we can also study the toxicity profile before going into the animal study, before going into the clinical study. You can also get to know the information of toxicity of that particular drug. Okay, so that's why AI is helping to study all these parameters: absorption, distribution, metabolism, excretion, including toxicity. You can also predict the toxicity of that particular molecule before starting your wet lab. Yeah, this is what how we are finding the toxic information of a drug. Okay, that is in silico toxicology. So we know in vitro toxicity, we know in vivo toxicity, but we can also do some in silico toxicology studies by using artificial intelligence, right? So these are the some of the molecules, recent molecules like HERG liability. So they predicted cardiotoxicity risk by using AI before going into the wet lab. Okay, so these AI models trained on decades of failed pharma data. Okay, so now this so many failures. Okay, these so many failures, and they will take that information, they will understand why they failed, whether any similarity is there in this particular drug or not. They will predict. So that will give more safety data, safety information. And then some of the recent trials, they used AI by by simulation studies, simulating drug pharmacokinetics and pharmacodynamics across diverse virtual cohorts. So we are doing all these pharmacokinetics and dynamics in silico itself before going into the wet lab. Okay, there are LLMs, there are some GPUs. Okay, so these will handle the big data, and they will design, they will give you the information whether these particular parameters are going to help or not. Okay, and the other thing is animal to human translation. So predicting human dose from animal data with far greater accuracy than the traditional allometric scaling. So initially we used to some of the example, I'll tell you here, insulin. Okay, so what is the source of the insulin? That is the animal source, right? People used to extract the insulin from the animal, before 1986. Okay, but some of the patients used to get the allergic reactions because that is animal insulin. It is not suitable for the human. But now, once the gene tech, they discovered the human insulin, they cloned human insulin to the recombinant E. coli bacteria. Now, E. coli is producing human insulin directly. There is no allergic problems. So that's how this animal to human translation is happening in a beautiful manner, right? And then AI also helping in preclinical imaging and pathology. So it is helping developing models, okay, and developing reference models. And just you put that reference model, you apply all the images, it will analyze the data. It will analyze the image data as well. And this particular work used to take so much of time in the olden days, right? That is especially MRI and PET scans. So all these image data we can analyze by using AI, artificial intelligence, right? And then so in phase 1, phase 2, phase 3, all these clinical trials, okay, so it used to take 3 to 4 years to get the data. And even from the regulatory agencies, once you submit the drug data, once you submit the ligand data, okay, so ligand data, then if you submit FDA or CDSCO, they used to take years to approve, because large data, they used to take time because they are not using AI, and people are sitting and they'll sit and look into each and every parameter. They used to take so much of time. But now the regulatory agencies also updated, they are also using AI, they are processing the data in a faster manner. So before AI, so 80% trials delayed by the large data. Okay, and now the scenario is completely changed. Everybody, even pharma industry and the FDA regulatory agencies, all are using data, and then it is easy now and to get the approval from the regulatory agencies. And then the other important thing is precision medicine. This is what I'm discussing, precision medicine. So same drug, same dose will not act on the all patients, those who are working with the same disease. Okay, so the metabolomics, the genetics are different from patient to patient. The CYP enzymes activity is different from patient to patient. So that's why now precision medicine will take place. So based upon your biological information, based upon your physiological biochemical information, and we need to give the drug. We need to give the prescription. So that is right patient, right trial. Okay, AI helping is right patient and right trial. And there are some important important wings of this artificial intelligence, that is synthetic control arms. So this is the important area of the drug discovery, and the artificial intelligence is playing a major role in finding out the patient data. Okay, so the historical patient data creates a virtual placebo group and reducing the number of real patients needed in the control arm. Okay, so we can minimize the patient use, because in the clinical trials, phase 1, phase 2, phase 3, we need all human volunteers, patient volunteers. Okay, we need all those. So if you use AI, so with the minimum number of groups, we can predict what are the parameters, then that can be used as the standards, right? So this is how we can AI help in the drug discovery, and especially in the clinical trials also. And then the other important thing is decentralized trials. Okay, wearable data. Now the smartwatch. If you see the example of smartwatch, so once you wear the smartwatch, it will give you heartbeat. Okay, it will give you oxygen percentage, SpO2, and it will give you BP also, right? So it will give the information. The once some of the, if a patient is a cardiovascular risk, if he is having cardiovascular risk, then you can wear a smartwatch. Then you can continuously monitor the blood pressure. Okay, so and some in nowadays we are also having the smart watches or smart devices that can tell you the blood glucose levels also, how much, right? So that is continuously monitoring, and it is now it became easy to take the medicine in a proper time. Okay, now and the some of the devices connected to the doctors. Okay, doctor is sitting somewhere, patient is sitting in his house. Okay, so now the doctor monitoring the patient parameters by sitting in the hospital itself, no need to travel. Okay, so by using wearable data, right? And protocol optimization and via simulation. So now protocols are optimized by the artificial intelligence. Okay, so the trial protocols are run in silico to identify bottlenecks. So it will identify the frequency and will identify the endpoint selection and dropout rates. Okay, before a single patient is enrolled. So no need to have a patient, but before getting the patient, and we can get the information of that particular drug action. Okay, so this is how artificial intelligence is helping in a beautiful manner in clinical trials. And coming to the pharma industry, okay, that is a healthcare system, but now what is happening in the manufacturing sector? Okay, so from moving from batch manufacturing to continuous manufacturing now. So just by using AI, you can go for continuous. So earlier, what they do, so they will manufacture one certain group of dosage forms, then they'll go for the check, then go to approve. But now AI helping us to continuous manufacturing. So you just manufacture, and AI continuously monitoring. It will check. Okay, it's continuous checking by continuous QC checking, and then we can go for the approval, right? So that is process analytical technology, PAT technology. The sensors will take the information whether the batch need to be approved or not. So that's how this is helping in the manufacturing also. So another, predictive maintenance and yield optimization. So in yield optimization also, this machine learnings and LLMs, they optimize the cell culture media and feeding strategies for maximum productivity. Inside the fermenter, how much energy is present, how much media is present. Okay, so whether is it's going to end, then the AI will it will get the information through the sensors, then it will give the immediate alert to the who is operating the machine, and they will feed the media. So this is how the yield optimization is also happening with the AI. And then supply chain resilience. So if the patient, suppose in the now the cancer, if we talk about cancer, so many patients are getting cancer now. So many people are getting, the cancer rate is increasing. So the demand is increasing. Then we need to come up with a solution in the same speed, the same quantity. The number is increasing. If you are the quantity, the research which you are doing is a slow phase, then it is going to become a problem. Okay, so when the demand is increasing, at the same time that demand need to be reached in a small time, if you are using with the artificial intelligence. Then post marketing surveillance, this is also important, pharmacovigilance now. So by using AI, so whoever may be reporting, if suppose in Jaipur, one patient is uh, suffering with the administering one drug. Okay, so that information uh, given by the patient, single patient, and then that will go to the manufacturing company, that will go to the regulatory authorities in a 1 or 2 days of time. Then immediately they can announce through all over India, so to stop this particular drug. Okay, by looking at the why it is happening, whether it is patient problem, whether it is drug problem, whether it is whether is it possible to occur same incidents in other patients also, that they will study everything. Once it is yes, it can happen to other patients also, then the regulatory agencies will immediately ban the drug from the market. So this is how the AI is helping the conveying the information from one phase to another phase. In each and every gap it is filling. Okay, so that is happening by the post-marketing surveillance, clinical trials, phase 4. And some of the other important platforms. Okay, this literature review. Okay, so this literature platforms helping the pharmacovigilance scientist and drug discovery scientist. Okay, so to monitor the ADRs, especially automated ADR extraction. So these NLPs, okay, they are trained in the adverse drug reactions. So this extracts the adverse drug reactions from unstructured clinical notes, enabling faster signal detection by FDA or EMA reporting, whether it is US FDA, then if it is India CDSU. So this is how the ADRs are monitored continuously, monitored timely. So if some ADR is reported in Jaipur and that is not reported to the FDA, that is not reported to the CDSU, then the same ADR may happening in other countries as other states as well. So now this LNPs making and communicating regulatory agencies, communicating manufacturing, pharma industry, communicating doctors in a faster manner so that the ADR is monitored. Okay, then coming to the some of the case studies, as I explained, I will tell you some case studies from 2020 to 2026. The new generation of AI, that is native companies. Some of the companies like Pfizer, okay, and Dr. Reddy's and Novartis, they already started using AI. Okay, now our Google CEO like Sundar Pichai, right? In a few years back, he said that by looking at the retina image, then they can identify the problems or disease are coming into that particular patient, whether they are going to face any cardiovascular disorders in coming days, whether they are going to get any metabolic disorders. That is non-invasive model. They're not taking any blood sample out from your body, they're not doing anything, just looking at the retina. If you sit in front of their computer, just they'll take your retina image and then they'll give you the information about your disease and about your physiological conditions, right? So this is how the AI is helping. So one of the case study here I want to tell you all of you, so that is in silico medicine. Okay, so multiple AI designed molecules now in phase 2 and phase 3 clinical trials, completely developed by using in silico. There is no intervention. There is no intervention, but right now they are in clinical trials. Okay, so they approved phase 1, they approved, and now they are in phase 2, phase 3. So first true proof of concept for fully AI driven drug discovery, right? So this is one of the example. And then the other example like recursion pharmaceuticals. What are these? These are the massive automated microscopy combined with AI to map cellular responses to thousands of perturbations and phenotypic screening at a scale of impossible of human researchers. So lakhs of images analyzed with a single AI model. So it is not possible with the human beings or human researchers. Okay, we can identify the cell structures also. Okay, and we can identify the cell response, how the cell is responding. Okay, whether it is inhibiting, whether it is exciting, whether it is suppressing. So that response also noted by the AI, and that will give the information to the researchers, which is not possible to the human researchers. And then some of the other examples like Excentia and Abcelera. So what they did, so they developed AI driven antibody discovery platform, so played a critical role in rapid pandemic response, identifying therapeutic antibodies in a record time. So these company developing antibodies in a short span of time, right? And then another company, Excentia, the first AI designed small molecule to enter into the clinical trials, that is DSP 1181, right? So this is the drug they are going to get into the market after getting approvals by using completely AI, right? So if you are familiar with AI, then you are going to get absorbed by all these companies like Eli Lilly. So these company using AI, in internal AI hub for target ID and clinical optimization. They are using artificial intelligence for the drug discovery. That is Eli Lilly, the one of the famous company. Then Novartis, as I said, Novartis also they started using complete AI platform for generative chemistry platform. Okay, they will develop the molecules by using artificial intelligence against the particular one receptor. Then coming to the Pfizer, by using machine learning models, they are using AI for clinical trial optimization and manufacturing. Then Roche, this is also one of the biggest top 10 pharmaceutical company in the world. They are using AI in diagnostics. Okay, so that's why if you have AI skills, then you are going to absorb in pharmaceutical industry in a short span of time, right? And then the other important, so data silo, the quality problems. So this is not the algorithm, it is the data. So the biggest massive giant data handled by the supercomputers, and they will give you the accurate information, right? And so the another important thing is, so deep learning models lack the transparency. So there are some problems. That's why you have to use this AI. So once again, I'm telling AI is a double-edged sword. Okay, this is the double-edged, two sides. They have edge. You must use in a proper way. If you use in a wrong way, it will harm. Okay, so that's why there are some problems you have to use. So it is lack the transparency, and medicinal chemists need to know why a model suggested a structure to trust it, and regulators need to know too. So just AI gave the molecule structure, you cannot blindly go. So the regulators and the medicinal chemist must to work on that particular structure, whether it is really work or not, whether the parameters are going to fit to the uh, particular receptor or not. Without interpretability, AI is not safe. Okay, so that's why you have to interpret with the basic knowledge. That's why AI is not going to cut off the jobs. It is going to give you the job, and it is going to fasten the drug discovery, right? And these are the some of the regulatory agencies like FDA, EMA. They are also working on AI. So they are going to give so many. If you go, if you visit FDA website and EMI website, they have given a beautiful AI models to useful for the drug discovery. You can use your projects as well, right? And yeah, these are some of the data privacy and patient consent, right? This is also happening by the AI. They are storing the patient data, right? And then yes, quantum computing in drug discovery. So classical computing is now nowhere is happening directly. AI is counting, and that is giving the chemical information, drugs information in a faster manner, right? So self-driving labs, if you have AI tools, if you know how to use AI tools, then you can start your project in a faster manner, and you can communicate with the researchers. And yeah, these are some of the applications are happening in the AI by using AI. So now coming to you, as you are a student in a graduation or B Pharm or PharmD or whatever the degree, so you must have some information, some soft skills to get absorbed into the industry. Example, so soft skills: cross-function communication, critical thinking, and asking the right biological questions to AI model. You don't uh, um, spend time with AI without knowledge, without proper target, without proper aim. Okay, and some of the techniques, skills you must know, that is basic Python and ML concepts. Okay, and single cell data analysis. Then you will get the now the current job market. So computational biologist, multi-omics. If you know the multi-omics, there are so many jobs offered by. Just now I said so some of the company names. They are offering jobs in AI. And then AI product manager, bridging the science and software in pharma. You must know the biological information. You must also know the computer information, okay, computer knowledge. And digital biomarker scientist, wearables and medical devices. Now the government of India almost they allocated 20,000 crores for this biopharma, okay, especially medical device and drug discovery, startup companies, right? So you can utilize all those things. And then cell therapy data scientist. Okay, so now we are working on CAR-T, gene therapy, and biologics. So to discover new treatment methods for the cancer patients. Now, so we are working on CAR-T. So we are going to formulate the genes with the nanolipid formulations, okay, nanolipid particles. Then we are entering into, we are going to work with the cancer patients. So that's how in a graduation level, you try to use the artificial intelligence and then try to learn some basic techniques, okay, how to see the protein structure, how to see the gene structure, okay, gene sequence, okay, and how to do the ADME with the software tools. So like that, if you know minimum basic skills in AI, then you are going to absorb by the pharma industry uh, in a short span of time. The future is yours. Okay. So AI transforms every step of the drug life cycle, right? And that it is going to involve in every stage from target identification to and drug discovery. So every stage AI is going to impact. So that's why so AI will reduce R&D cost and freeing the resources from rare diseases, antimicrobial resistance, and complex solid tumors also. We are going to target even solid tumors as well, right? Now the tools are ready, the data is growing. The next breakthrough will come from this room itself, from your classroom itself. If you know how to handle data, if you know the tools of the AI, then you are going to become a great scientist in some of the worldwide companies of pharma, right? So with these small words, I thank the management, and a special thanks to Ankita madam for inviting me to deliver the lecture, to interact with the all pharma students. I am very happy to interact with young minds. So any questions you can ask. You can for the some internship we provide in CCMB. So you can apply internships also for Form D students and B Farm students. Okay. So there are some summer training programs in national institutions. It is free of cost. You go through the website of CCMB and CDFD. Okay. If you are really interested, and you can apply the internships and you will get, but that is only in summer. Okay. You have to apply, you have to compete. So national wide applications will come, and if you are selected, yes, you are in.
Okay, so participants, if anyone has any kind of doubt, you may ask. So the session was very interesting, and here is an opportunity also provided by Dinesh sir for the internship program, which may you participate. So as we come to end of this insightful session, I would like to extend my sincere gratitude to Dr. Niradi Dinesh sir for sharing his valuable knowledge and expertise on AI in drug discovery and development. Sir, your insight have really broadened our understanding on the emerging role of AI in pharmaceutical research and will definitely inspire our students and academicians to explore new possibilities in the field. So, on behalf of Pacific College of Pharmacy, Pahir University, thank you once again for your valuable time and being a part of Gurukul Pacific. I would also like to thank our respected President Sir, Dean Sir, and faculty members and all the students for joining and making this session meaningful. Thank you everyone for the enthusiastic participation. Have a great day, have a wonderful day ahead. Thank you once again. Thank you, sir.
Thank you.
जी sir, we will definitely going to invite you in our college also for the offline session and training
Sure, sure,
programs. Thank you
sure,
so
sure, sure.
much, sir.
Yeah, okay.
Yeah,
Yeah, thank you, madam.
you may all leave the meeting now. Thank you so much.